From the Library of Congress in Washington, D.C.
Good afternoon. Welcome to our program, The Origins of the RNA World, a collective oral history. We're glad you're here. We're glad the Metro shut down yesterday, not today. This is a good thing. I'm Dan Torello. I'm on staff of the Kluge Center. And before we begin, I want to remind you of a couple of items. We're recording today's conversation for future placement on the library's YouTube and iTunes channels. And also, please turn off any cell phones or any other devices that might interfere with the conversation. This afternoon's conversation is being moderated by Nathaniel Comfort. He is the Bloomberg NASA Library of Congress Chair in Astrobiology. I'm going to introduce him in just a few moments, but first a few words about the Kluge Center and about the Bloomberg program as a whole. The Kluge Center was created 16 years ago, thanks to a generous gift by philanthropist John W. Kluge. We have two primary roles. The first is to create a scholarly community of approximately 100 scholars every year. These are senior scholars, postdoctoral fellows, predoctoral fellows, all of whom contribute to a vibrant community here on Capitol Hill and in the library. The second function is to administer the Kluge Prize. This is awarded by the Librarian of Congress. It's a $1 million award that recognizes lifetime achievement in the study of humanity. It was last awarded to philosophers Jürgen Habermas and Charles Taylor this past summer. So within the Kluge Center, the Bloomberg Chair in Astrobiology is the result of what has been a unique and rewarding collaboration between NASA and the Library of Congress. The chair takes its name from Nobel Prize winner Barry Bloomberg. He was the founder of the NASA Astrobiology Institute and also a founding member of the Library of Congress Scholars Council. Bloomberg envisioned the creation of a chair in the Kluge Center that would focus on the humanistic and societal impacts of astrobiology. The idea was to connect scientific discoveries in the field of astrobiology with the best thinking in the humanities and social sciences. And astrobiology as a field of inquiry in particular is one that allows for the big questions, things like investigations on the origins of life, about which we are going to hear more about this afternoon. So placing scientific knowledge within the context of historical and philosophical thinking gives us the chance to consider questions of meaning and of value that are important to us as human beings. Nathaniel is the third chair to be in residence. Our first chair was David Grinspoon, who I see sitting here in the back. He researched a book on the Anthropocene. We then had historian of science Stephen Dick, who wrote about the potential societal impact of the discovery of microbial or complex life beyond Earth. And this year we've been thrilled to have Nathaniel with us since October, and he is looking at the history of the genomic revolution and origin of life research. In addition to holding the Bloomberg Chair here at the Kluge Center, Nathaniel is professor in the Department of the History of Medicine at the Johns Hopkins University. He has published scores of articles and several books. I mentioned just a few of the more salient ones: The Science of Human Perfection, How Genes Became the Heart of American Medicine, published by Yale in 2012. The Tangled Field, Barbara McClintock's Search for the Patterns of Genetic Control, published by Harvard in 2001. He's also the editor of the volume, The Panda's Black Box, Opening Up the Intelligent Design Debate, published by Hopkins in 2007. He writes extensively for The Atlantic, The Nation, the New York Times, Book Review, and other outlets. He also blogs, so you can look this up. His blog is genotopia.scienceblog.com. So we've been thrilled to have him with us this year, and we're grateful to him for bringing such a distinguished group of panelists to the library today, and we look forward to a fascinating program.
Well, thank you, Dan. It's a real pleasure to be here, and I would just like to begin by with a word of thanks to the Kluge Center. This is an absolutely marvelous and a unique place to be. I'll just tell you how great it is. When I got my acceptance letter, it had a line in it saying that we expect you to protect your time for research and scholarship and reflection. Now, how rare is that? I mean, in these days. So this is a wonderful place, and I'm delighted to be here. And I'm particularly thrilled to have this distinguished panel of guests with me today to talk about the RNA world, which is an important aspect of research on the origin of life. And I'm just going to introduce them briefly in alphabetical order. First, W. Ford Doolittle, who's from Dalhousie University in Halifax, Nova Scotia. He got his PhD in biological sciences from Stanford. He then did postdoctoral work with the distinguished scientists Saul Spiegelman and Norm Pace. He then joined the Department of Biochemistry and Molecular Biology at Dalhousie in 1971, and he's been there ever since. He's won a Guggenheim Fellowship. He's a member of a fellow of the Royal Society of Canada, a member of the National Academy of Sciences, and on, many distinguished honors. And his research has been on the molecular genetics of microbes and things like lateral gene transfer, instead of hereditary transfer, transfer of genetic material across species, selfish DNA, gene structure, particularly introns, about which I'm going to say a little bit in a moment, and the tree of life. And one thing that's really quite distinctive about this scientist is that he right now has 2 postdoctoral fellows who are philosophers of science. So he really bridges the sciences and humanities, which is something that I find really appealing. George Fox is the Moores Professor of Biochemistry and Biology at the University of Houston. He got his PhD in chemical engineering from Syracuse. He's the co-discoverer with Carl Woese of the archaea, one of the 3 fundamental domains of life, which really overturned the way people thought life evolved. He is a fellow of the International Astrobiological Society, member of the American Academy of Microbiology, a fellow of the American Academy of the Association of Science. And his research has been on the evolution of the machinery of genetic translation, how information moves from nucleic acids into amino acids and forming proteins that form the enzymes and interesting structures in the body. Ray Jesland is the Emeritus Professor of Biology and former Vice President of Research at the University of Utah. He's a Howard Hughes Medical Institute investigator. His PhD was from Harvard, where he studied with James Watson. He did postdoctoral work in Geneva, Switzerland, and joined Cold Spring Harbor Laboratory in 1967, staying there until 1978, when he joined the University of Utah. With Ray White, he began a new and now very distinguished Institute for Human Genetics. And since 1972, he has collaborated with John Atkins on the phenomenon of genetic recoding. He is the editor of the volume RNA World, so he wrote the book, or at least edited it, and which is now in its 4th edition. So it's really the sort of Bible of this field. So it's really a delight to have him. And Walter Gilbert is the Carl Loeb Emeritus Professor at Harvard. He has a doctorate in physics from the University of Cambridge and was appointed to the Harvard Physics Department in 1959. He then switched from physics to biology, and he ran a lab jointly with James D. Watson of the double helix fame. He won a Nobel Prize in chemistry in 1980 for the invention of a method of DNA sequencing. And he's a pioneer in biotechnology. He founded Biogen, one of the first biotech companies, if not the first. He oversaw the development of a number of groundbreaking products, including alpha interferon and the hepatitis B vaccine. And he also co-founded the large biotechnology company Myriad Genetics. Other honors besides the Nobel Prize include Louisa Gross-Horwitz Prize, the Gairdner Award from Canada, the Lasker Award, National Academy of Sciences. He's a foreign member of the Royal Society, British Royal Society, and on and on. His research has included the identification of messenger RNA, the lac repressor, fundamental component of the lactose gene in bacteria, model system. Pioneering work in recombinant DNA, the first genetic engineering, including the expression of insulin in bacteria. He was a founding, a pioneer member of the Human Genome Project, and he coined the term RNA World. So, it is an absolute thrill to have these gentlemen here with me today and to get into a conversation with them. And before we start that, I just want to give a few remarks just to make sure we're all on the same page, just introduce you to the idea of the RNA world. Okay? So, I can't see, oh, here we are. So, what is the RNA world? Well, beginning 1953, James Watson, Francis Crick, aided by Morris Wilkins and Rosalind Franklin, of course, solved the double helical structure of DNA. Now, the double helix, the shape of the molecule usually gets all of the credit, but the really interesting thing about it is those bars on the ladder crossing the spiral staircase, because those symbolize the nucleotide bases that pair up. So it's really two half spiral staircases, right? Each specifying another nucleic acid, and they realized that in the sequence of those bases was genetic information. Okay? So it was in the DNA that the information was held for making proteins. DNA information goes to protein. The A's, C's, G's, and T's of DNA turns into the amino acids that are strung together to form proteins. Proteins are crucial molecules in the cell. They do all sorts of work. They are enzymes, most importantly, but they're also structural and the basis of neuronal transmission and structural elements in the cell and so forth. Crucial components of the cell. And in 1956, 3 years after the double helix, Francis Crick coined what he whimsically called the central dogma of molecular biology, and Watson drew it out in this cartoon, which says a number of things about the flow of genetic information. This was really the first time people began to think about genetic information. And the important thing that most people remember of the central dogma is this simple phrase: DNA makes RNA makes protein. Okay? So, what's DNA versus RNA? They're very similar molecules. There are really only two differences between them. One of the 4 bases is different. RNA uses uracil instead of thymine, and you'll see there that there's on the ring there's one difference, an OH instead of an H. Those are the only differences between the two molecules, but those two slight differences make huge differences in the chemistry. And DNA, of course, forms a double-stranded molecule. RNA is usually single-stranded, and that means it can bend around and form into all kinds of different shapes. Right? It's also much more reactive than DNA, and those two facts become important in the RNA world. Okay, so the DNA makes RNA makes protein through two processes, one called transcription, where an RNA molecule, messenger RNA, which Dr. Gilbert was a key figure in identifying. And then that messenger RNA is translated into a sequence of amino acids that makes a protein. Okay? And as I said, proteins are ubiquitous in the cell. They form hair, nails, muscles, nerve cells, and most importantly, enzymes. There are a couple of, and that translation process is really sort of the heart of what we're going to be talking about today. Translation occurs in structures called ribosomes. So the messenger RNA is read off the DNA, and then it moves outside the cell nucleus to the outer part of the cell. And in the ribosome, it is pulled through like a tape through a tape recorder, and it is read off, and the sequence of those nucleotides specifies the sequence of amino acids. So this building chain of amino acids then forms the protein. Now you'll notice a couple of things here. One is that there is an awful lot of RNA here. You have the messenger RNA, ribosomal RNA. The ribosome itself is made of RNA, mostly. And then, if I can get one more, and then transfer RNA is the molecule that actually attaches to the amino acid and brings it to the building chain. So there's a lot of RNA in this part of the cell. Okay? People very quickly began to notice this fact, and in 1962, one of the pioneers of molecular biology, Alex Rich, gave a paper on the origin of life. So this is just 9 years after the double helix, and in fact was the year that Watson, Crick, and Wilkins won the Nobel Prize. And he said the hypothetical stem or parent nucleotide molecule was initially an RNA-like polymer, which was able to convey genetic information as well as organize the amino acids into a specific sequence to make proteins. Okay? So RNA does most of the work of that process. Right? And in 1965, just 3 years later, two other scientists made similar realizations. J.B.S. Haldane said that life, by which he meant indefinite replication of patterns of large molecules, can be based on RNA without DNA. And Fritz Lippmann, at the same conference, said that the DNA probably evolved later than RNA. So there was a time in which you had RNA and proteins and not yet DNA. Okay? So back to this diagram here. Another thing that we can notice about this is that there are enzymes or proteins involved in each of these processes. So none of this, although there are RNA molecules involved, none of this can happen without enzymes, without proteins. All right, so these things are specifying proteins, and you need proteins to do it. So that leads to a kind of chicken and egg problem, right? Where you have genes that store information that encode proteins that make enzymes, and you need those enzymes to catalyze the reactions to copy the genes. So how do you get out of that problem? The RNA world is the way you get out of that problem. Okay? And several people noticed this in a speculative way in the late 1960s. Carl Woese said that proto-RNA could have been the proto-ribosomal RNA could have been the original genome. And Francis Crick the next year said that possibly the first enzyme was an RNA molecule with replicase properties, the ability to copy other RNAs. Thus, a system based mainly on RNA is not impossible. And the same year, his colleague, Leslie Orgel, asked, could polynucleotides of RNA with well-defined secondary structures, folding, act as primitive enzymes? Could they be more than just information storage molecules? Could they actually catalyze reactions too? That would be a way out of the chicken and egg problem. So the answer is yes, but hang on one sec. First, I want to tell you about a couple of things that are going to come up in the conversation. One is as I mentioned earlier, the discovery of the archaea, this brand new group of single-celled organisms that George and Carl Woese recognized through sequencing RNA, ribosomal RNA, were different from the bacteria. And so this created a lot of discussion about what was the original branching structure in the tree of life. Okay? And the same year, 1977 was a big year for origin of life research, Phil Sharp and his colleagues at Yale and Rich Roberts at Cold Spring Harbor and his colleagues independently discovered that genes come in pieces. In higher organisms, eukaryotes, organisms with a membrane-bound nucleus that contains the chromosomes, have genes that can be segmented. Okay? They can, and when you go through the transcription and translation process, those middle bits between the segments get spliced out, and the segments join together to form the final RNA that gets read into the protein. So that's really odd. Genes come in these pieces. Walter Gilbert wrote an article the next year called Genes in Pieces. And in that paper, he named those segments exons for the segments that are part of the genes, and introns as the segments between the exons. So they're in between the exons. Okay? And he and Ford Doolittle, over the next few years, got in a lively discussion about which came first, when introns were invented evolutionarily. Were they part of the very first organisms, or did they get invented later as the eukaryotes evolved? So this was a very important discussion in origin of life research that I want to talk about more later. And then finally, the discovery of RNA enzymes. Crick and Orga were right. RNA can act as an enzyme, and that was found first by Tom Cech in 1982, and then Sidney Altman at Yale found that a molecule called RNase P that he'd been working on for years also had, the RNA also had catalytic properties. So RNA can act as both a catalyst and an information molecule. So that breaks the chicken and egg problem, right? So this is kind of like the old Saturday Night Live sketch where you have the the shimmer, which is a floor wax and a dessert topping all in one, right? The next, just a couple of years later, Walter Gilbert wrote an article called The RNA World, and this was the coining of the term, okay? And we're going to talk more about that. I just want to put that up there so you have a sense of the dates, okay? In 1987, Cold Spring Harbor held a meeting on RNA catalysis that involved a lot of discussion about the RNA world. And in 1993, Ray Jesland and John Atkins edited a volume of a collection of papers called The RNA World. And as I said, it went through multiple editions. I forgot to put up the fourth. What year did the fourth edition come out? 2011. Okay, so this is still a very vital and ongoing project. All right, so that is a quick introduction to the RNA world. Now I'm going to sit down. I'm going to mostly turn it over to them. Thank you. Okay, just wave your hands or something if you can't hear or can't see. I'm trying to get out of the way, but I also want to make sure that I'm in conversation with my guests here. So, with that, let's begin with evolution. Theodosius Dobzhansky famously said that nothing in biology makes sense except in the light of evolution. But the early generation of molecular biologists were often criticized for not taking evolution sufficiently seriously. One of those critics, at least in my reading, was Carl Woese. What do you think? Is that criticism fair? To what extent did the RNA world emerge from evolutionary thinking? I mean, from thought based on serious reading of evolutionary literature.
I think that's an unfortunate question. I do agree. The early generation of molecular biologists did not, for the most part, didn't think of evolution. The discovery of the DNA sequencing in '76, suddenly made it possible to look at the genes of many organisms. And one of the first things that happened as molecular biologists did this was they discovered that the genes in Drosophila were like the genes in plants and things like that. And suddenly, evolution swept the field. I remember a Gordon conference in which, in fact, this sort of suddenly became a fantastic element in people's thinking. Those of us who had a slightly more genetic background were more conscious of evolution, but the general field of molecular biology was not. I think the RNA world image is deeply based in a notion of evolution. The point of the paper that I wrote in 1986 was twofold. It picked up from arguments that had been made, it picked up from the discovery of the RNA enzymes that had been made and reiterated just before that. And it picked up from an older notion from the biochemists that RNA was more primary than DNA. And the biochemistry of that is that all of the DNA structures, the DNA sugars and the DNA, often the DNA bases and nucleotides, are made from RNA precursors. So if you look at the biochemistry, you would not say DNA is separate in any way or primary in any way. You'd say, oh, the chemistry suggests that RNA is the thing that you made biochemically, and from that you would later make DNA. People, knowing that, thought of a RNA protein world. RNA as a genetic material, machines in the organism, and that genetic material can be used to dictate the sequence of amino acids. And amino acids were thought of as, oh, the interesting things in the world, the enzymes. And the protein chemists ignored the nucleic acids intensely. They thought everything interesting in the world was in the proteins. It makes very interesting meetings in which the protein chemists meet separately and the nucleic acid chemists meet separately, and they never like to talk to each other, and the protein chemists totally ignored nucleic acids.
Nobody could translate between the two languages.
But the discovery of the enzymatic activity of RNA made me think that maybe RNA could be entirely play the role of all of the enzymes. And so the paper I wrote in '86 makes actually two statements. One, it suggests maybe RNA enzymes could in fact do all of the necessary activities for a cell to exist, including the copying of RNA itself. And therefore you could imagine evolution as beginning with a small RNA molecule that develops by random structures, random association of bases, the ability to copy itself. And once it can copy itself, it can make it makes more copies. But in fact, since it's a biological process, it makes copies which would also have errors, and so it can evolve. It can make random changes, and those random changes can be acted upon by natural selection to eventually produce better molecules. Second part of that essay, however, points out that because one of the RNA enzymes that was then known at that time was an intron, the region inside an RNA molecule that could splice itself out, I could make the argument that that activity clearly could be used not only to splice introns out of the RNA, but by combining two of those structures, develop a structure that could move an RNA piece around from one molecule to the other, inserting it like a transposon, splicing out the rest, and creating a novel RNA combination. And that's a very powerful evolutionary tool. We call it recombination when it occurs at the DNA level, but in fact here was a model that clearly could happen at the RNA level. And so I could immediately in that paper suggest two things. Maybe there could be an RNA-based organism completely using RNA as enzymes, and the evolutionary processes would be able to shuffle the pieces of RNA around to make novel pieces again. The original intron-exon idea you alluded to, yes. The critical element of that idea was an evolutionary argument. It was not just the biochemical argument that you had on the DNA long regions that we call introns that are spliced out of the RNA molecule to produce small pieces together that code for pieces of the protein. But the evolutionary argument is that by spreading those regions apart in the DNA, you would increase the recombination rates between those regions. And hence you could more rapidly create protein structures and evolve them to better function by recombination and by shuffling the pieces of the protein. So in fact, both the intron-exon idea is conditioned by a vision of evolution, and the RNA world idea is also conditioned by that vision of evolution. That paper, in fact, finally includes that sort of a hidden definition of life as that thing which can, by replication, mutation, and recombination, that is by changing, random changes, can evolve, and thus can be operated upon by natural selection to produce ever better functioning.
Okay, thank you. I think that was a very fortunate question in that case. I can now throw away half my questions, but we're... Ford, I think you might have something to say on this.
Yeah, well, I think your initial question involved the phrase serious reading of the evolutionary literature or something like that, and I think most molecular biologists did not seriously read the evolutionary literature, probably still don't. Mostly evolution is something you talk about in the last paragraph of the paper, I think, and over a few beers kind of thing,
right.
Because I think there was a general feeling amongst the molecular biological community that evolution obviously was important, but also there was nothing you could really say about it, so you could just say whatever you wanted and everybody would accept it politely. And I think
I think that's right.
I think you were right that 1977 was a very important year for some of us, and I want to go back a little bit about the intron relationship to the RNA world, because I was in Wally's lab on sabbatical at that time, and Wally came back from Switzerland, I think, having heard for the first time about the introns and the immunoglobulin genes, I believe, and gave, there were lab meetings once a week in Wally's lab with very strong tea, as I recall, and he presented
It was tea and not something stronger.
It was. And Wally presented the exon shuffling idea, which I thought was a brilliant way for things to evolve more rapidly in the way that he just described. But I had one concern about that, which was, I believe, thinking the way that most people thought in those days. We imagined that higher cells, eukaryotes, animals and plants, evolved from bacteria, and bacteria don't have introns, and eukaryotes, higher cells, do have introns. And so that sort of implied that these relatively advanced cells would suddenly take on these interruptions in all their genes just so that they could in the future do a better job of evolving, which seemed not something anybody who had done any serious reading of the evolutionary literature would be willing to accept as possible.
You're not quite fair.
I'm not blaming you for that.
No. I was going in large. At that moment, we knew of the introns only in more complicated genes. And so the first suggestion, I learned about the introns and exons from the people who discovered the splicing in the Cold Spring Harbor meeting in probably May or June that year. And in my laboratory, we had a sequence of a immunoglobulin gene, which actually had an intron. We had just sequenced it. We didn't actually know what that was, but it was an intron. So when I came back from Switzerland, having thought about these ideas, I suggested that eukaryotes uniquely use the greater evolutionary speed of recombination within introns to produce the great explosion, Precambrian explosion of the eukaryotic genes. And that's the background for your suggestion.
And that's what I objected to.
Right.
Because if we believed, which I think 99% of the people believed at that time, that eukaryotes came from prokaryotes, then they would have had to have taken on the burden of introns, and 10 introns on average in every gene, in order that several million years down the road, they'd be able to do these wonderful evolutionary things, which seemed kind of anticipatory and not the kind of thing. Evolution really can't look ahead. So that was my reaction to what you presented. I don't think you actually implied that, but it was my reaction. And I also happened to be privileged because I had worked in Urbana, and I knew Carl Woese, and I knew George, and I knew about this almost simultaneous publication of the three-domain view of life that George and Carl Woese had been working on, that in their view, eukaryotes and prokaryotes were not evolved. I mean, it wasn't that eukaryotes evolved from within the prokaryotes. They evolved separately from some more primitive ancestral form. So then I thought, well, actually went home and had a bottle of scotch and wrote overnight that piece. and I thought that, you know, it'd make more sense if introns were actually present in the very beginning. And then as evolution proceeded, prokaryotes lost all the introns, but eukaryotes retained them, and this gave them the evolutionary ability to become complex organisms like ourselves. And prokaryotes haven't done that, although they've done a lot of other wonderful things, and they've gotten much more streamlined. We used the word streamlined.
Streamlining, yes.
And so I showed that to Wally, and I think he thought, oh, that was interesting.
I thought it was a very good idea.
Yeah, thank you. And and then I got in the mail a paper from Jim Darnell, who had been thinking along those same lines.
Was it Yale?
He was Rockefeller at that time.
Rockefeller, right. Yes.
You're putting everybody in.
I'm sorry.
Yeah.
A lot of people were there.
Well, a lot of people were not.
So this.
So sharp wasn't it?
So I think there were several things that came together at that time. There was a belief that several of us had, and a guy named Darrell Rainey had been articulating before that, and I think both Jim Darnell and I had this kind of gut feeling, and I got it just from teaching molecular biology. You know, you teach about bacterial cells, and they're all so elegantly designed and quick and easy, and then you look at a prokaryote and say, oh my God, what a mess. You know, a eukaryote, what a mess. How messy is the molecular biology of higher organisms compared to bacteria? So one way to respond to that is to say, well, maybe it's primitive. Maybe they just never got over the beginning, and, you know, the beginning was a messy time, and eukaryotes are still messy. So that was part of that. And but I think when Jim Darnell and I were thinking about exon shuffling, and I think when Wally was thinking about it at that time, we imagined it was a DNA process. I think we imagined that the genomes which were putting together their genes from exons were actually DNA genomes. Mm-hmm. And then,
but certainly for the eukaryotes.
Yeah, and then when Tom Check reported the splicing of introns, it occurred to several of us simultaneously, and to Darrell Rainey a little bit before me anyway, that you could just recast that whole theory in terms of the RNA world, which is what your '86 paper did. Mm-hmm. That's my.
Well, you know, you published the introns early argument, and I found it sufficiently attractive that I adopted it.
That you tried to prove it.
And worked on it extensively. Yeah. I still find it very attractive.
Do you believe it?
Certainly.
Oh, okay. I don't believe it.
I don't believe it?
No, you recanted, but I...
Yeah. Oh, well.
George, Ray? George?
Well, I was reflecting on the comment regarding the role of RNA in the earlier times and the importance of RNA. And I remember teaching classes, and I was teaching on protein synthesis, and I would teach the students about ribosomal RNA, which was called small r, capital RNA, ribosomal RNA. And there, of course, there's a gene for ribosomal RNA, you know, there's genes. And so there's something called ribosomal DNA, rDNA, which codes for the ribosomal RNAs, okay? And then I come to learn from my students that no, I didn't know what I was talking about. rDNA means recombinant DNA, okay? Because these people in the cell biology, et cetera, they thought RNA was completely and totally just irrelevant. And so, I mean, that's the way it was for many, many years until this RNA hypothesis came along. Now, about the RNA hypothesis, there's still a lot of ongoing controversy because there's two aspects to it. One is, where did the RNA come from? So you need to have the RNA come from some kind of prebiotic chemistry, presumably. And so it's proven to be rather difficult to define where the RNA comes from. And so one of the alternative theories is that, well, by the way, DNA was not the original genetic material. RNA was the original genetic material, not DNA. So DNA replaced RNA as the genetic material. Therefore, maybe some other nucleic acid was the predecessor of RNA, and that predecessor would therefore be easier to somehow make in a prebiotic world. But in any event, somehow, if you're going to have this RNA world, the RNAs have to come, there has to be some mechanism for producing the RNAs. And there's not, although some progress has been made, actually, there's not yet a truly convincing argument of where the RNA came from to create the RNA world. Now, the other problem that maybe is easier to solve is the problem of the replicase. One of the key components of the RNA world, or the full RNA world, is the idea that you have a replicase. And so people have sought for many years to find an RNA that could replicate another RNA. And again, some progress has been made, but the systems involved actually seem quite artificial, and they're relatively complex. So in my case, I've come to propose what I call an abbreviated RNA world, because the thing which ultimately will terminate the RNA world is the presence of proteins. And Wally actually pointed this out in his original article in 1986, but once you have protein synthesis, the RNA world quickly will evolve to become an RNA protein world. And so our feeling is that, in fact, the first thing the RNA world successfully does is create the protein synthesis machinery. And so therefore, you never reach the point where you're having RNA enzymes catalyzing various metabolic pathways and stuff like that. Because the original RNAs end up being involved in creating proteins. And so that's sort of where things are right now. There's two mysteries: where the RNA come from, and how do we find a self-replicating thing. And so people are very worried about both of those things. The peptide synthesis idea, the proto-ribosome idea, gets rid of the second problem, doesn't solve the first.
So could you say that one of the functions of RNA early on was essentially to make itself partly obsolete?
Well, not on purpose.
By the way, that's an anti-evolutionary argument.
Yes, definitely is.
That's something you can't select for.
No.
See, the problem with that, the advantage of the RNA world model, full model, is that because there is a replication of RNA, replication of the enzymatic structure that's making more RNA, the structure can evolve and can be selected for, and better forms can arise. The problem with the proto-RNA image that you created is there's no replication in it.
Right.
And therefore, there is no use of the evolutionary forces to make better forms. And so it has to arise by accident in its final form before it can actually even begin to work. Whereas the RNA world model, in the form that I suggested, at least can begin with that first enzyme that can copy itself and then go on to better things. And the first one can be very primitive, but because it's copying itself and evolving, it can get better and better. Still have the problem, we don't yet have a full biochemical model for how an RNA molecule can be copied. I think the longest sequence that's been copied is 5 or 6 bases.
It's better than that.
Close to 100.
Okay, close to 100. Well, by the time you get close to 100, we're getting there.
But not
the paper
itself.
came
But
out
can't
this
copy,
year. It's
can't copy itself.
Closer to 200.
You know, it finally gotten to 100.
Oh, yeah.
I mean, at the time of the RNA world suggestion, there was nothing.
Yeah.
You know, it hadn't copied one base, and people worried about what the precursors were. So I'm encouraged by getting to 100 dramatically.
Yeah.
The other problem is the one that's mentioned, which I think there's still no solution to. How do you actually, in a prebiotic form, get to those RNA precursors?
Right.
Although progress is being made, so far there's nothing that's fully definitive. Because my confidence is both of these problems will get solved, and sooner or later we'll have a test tube with little RNA molecules running around making better copies of themselves, and we can try the whole thing all over again and see where we end up.
We might need it, too.
Rewind the tape, as Stephen Jay Gould said, right? Okay, so all of this complicated machinery, this is like an engine. It takes energy to drive this. None of these are thermodynamically favorable, right? Copying RNAs and coding for proteins and stuff. Where does the energy come from to make this happen?
Well, you're asking seriously?
I'm out.
Okay, in order to make a polymer, or to make the RNA polymer, the enzymology we use currently is an enzymology based on triphosphates. So I have to make it. I make a triphosphate. I break off a diphosphate, and can polymerize the rest.
A triphosphate of an adenine or a nucleic acid, right?
Of a nucleotide.
Nucleotide.
In order to make that process actually work monodirectionality, I have to get rid of the diphosphate, and I've got to eliminate that. Otherwise, the process is in equilibrium, and I can't actually synthesize molecules.
Right.
So I have to have a source of energy to make what we like to call high energy compounds.
Right.
That can be solar energy. It could be any. One could conceive of any number of sources of that energy. But I then have to finally make polymers. That takes energy. In order for the replication to continue to work and evolve, I've got to sort of I've got to freeze entropy. I've got to prevent the general degradation. I have to have a source of energy coming in. I have to have some process that makes complicated structures, which I can do using that energy, and then I have to have some process that is evolution to keep the structures going. I do have to have energy.
Yes.
I need something that provides multiplication of molecules.
Right? Oh, okay.
You're happy.
I'm happy. You're happy. Okay.
Happy.
Another topic that interesting, we talked a little bit about introns. About the same time, several of you, Francis Crick and Leslie Orgel once again, and Ford Doolittle, also published articles in the same issue of a journal, Journal of Molecular Biology. Both articles noted that they were inspired or spurred by a recent book by Richard Dawkins called The Selfish Gene, which came out in 1976. Some of you may have read it. And they wrote about what we would call selfish non-genes, if I may. This is in 1980, so right around the time that a lot of this exciting stuff is happening, split genes, the archaea, and so forth. So both the authorship and the timing suggest connections to the RNA world and to these discussions about introns. No?
That was in Nature, by the way.
It was in Nature?
Right.
Excuse me.
JMB is much too slow to publish.
Okay, fair enough.
I don't think there was actually much connection because, I mean, the notion that introns might be transposable elements didn't really come along yet. I think Tom Keller-Smith might have been mooting it at about that time, but most people didn't pay a lot of attention to what Tom said much of the time because he says so much.
This is Tom Cavalier-Smith?
Right.
Yeah.
So I think the selfish DNA papers that Leslie and Francis and I and Carmen Sapienza published simultaneously, thanks to the grace of Francis and Leslie, actually, we're pretty much coming out of having read Dawkins' book and being bemused by the fact that people were talking about the evolution of bacterial, the transposable elements, in that same anticipatory way, which I always react against, which is that these things exist so that in the future the cell will be better off, and that just doesn't make sense.
The important point is that evolution does not have foresight.
Does not look ahead.
Right.
And furthermore, large amounts of eukaryotic nuclear genomes were now known to be consisted of repetitive DNA, which was, at that time, I think, not quite yet realized that it was mostly transposable elements. But now we know that 45% or more of our genome is made up of transposable elements, which Orgel and Crick and Sapiens and I argued were simply parasites. They're pieces of DNA that can multiply themselves. Then, when the intron theory became more developed, and it was becoming apparent that there were no introns in bacteria, and that it seemed and that we knew that there were some group 2 introns, which now Phil Sharp had said were likely to be ancestral to the spliceosomal introns, the complex introns that eukaryotes have. Then Tom Cavalier-Smith's theory, which was that introns came in as group 2 introns from mitochondria, part of the mitochondria which are in the symbionts within the eukaryotic cells, that was the origin of the introns that then went wild in the nuclear genome of eukaryotes. That's what I think most people, with the possible exception of Wally Gilbert, now believe. So that was the connection, but it wasn't connection through the RNA world, it was connection through, there was no RNA world part to that.
Okay. Is there a partial exception to Wally Gilbert?
Yeah.
Okay.
I think you believe that some introns are late too. I don't think you probably have a all introns early view.
Well, yes, a few may be late, but that's,
a few, okay.
that's not very much.
So Wally's a holdout on this one.
Okay. And so would you say there's consensus around the idea that introns were present in the first genomes, the first RNA genomes?
No, there's a consensus that introns were present in the first eukaryotic, the last common eukaryotic ancestral cell. Was replete with many introns, supposedly. Most people think that the only introns that were ever in prokaryotes were simpler introns, in particular the group 2 ones, which according to a theory of Phil Sharp, which had the lovely title of 5 Easy Pieces, a group 2 intron leaped from the mitochondrial genome of an invading bacterium into the nuclear genome of the host, fell into 5 different parts and spread either before or after falling apart.
What was it doing in the mitochondrial genome of that invading bacterium?
Well, group 2 introns are mobile transposable elements in bacteria too. They're just not. They're restricted in where they can go because they will interfere with translation in a prokaryote. So there are not that many of them. But once they were into the nucleated eukaryotic cell, they could go crazy. Okay. That's the theory.
A little background that plant cells, of course, have chloroplasts, which are the photosynthesizing elements that give them their energy. And animal cells have inside them mitochondria, which are the little engines that provide energy for animal cells, right? And the idea is that those were actually little microbes themselves at one point early on, and that they were engulfed by another cell.
Sorry, yes, I was going pretty fast there. Yeah. That is perhaps the only thing that everybody in the field of molecular evolution would believe, which is that mitochondria descend from bacteria which were engulfed by a cell that didn't have mitochondria and deriving therefore the benefit of aerobic respiration, and that chloroplasts in plants derive from what used to be called blue-green algae and are now called cyanobacteria, a kind of bacterium, which and those were engulfed by those organisms that became plants to provide the benefit of photosynthesis.
But that's after the eukaryotic cell.
That would be, well, now there's a big debate there as to whether, well, the chloroplast definitely after, whether or not mitochondria was after the development of a complex eukaryotic cell, in which case, according to the theory of Tom Cavalier-Smith, the introns came in with that mitochondrion, or whether the mitochondrial invasion was at the very beginning of the formation of eukaryotic cells, is a huge debate, which if you're going to talk to Bill Martin next month, you'll hear about.
That's right. But this has to be before the plant-animal divergence.
Oh, yeah, yeah, yeah.
Okay, yeah, well before plants and animals split off. This is the common ancestor.
There's one possible clarification some in the audience might.
Yeah.
When in the bacterial cell, when the messenger RNA is made, the ribosomes attach to it immediately and begin translating it. So there's no opportunity to remove the intron. Whereas in the eukaryotic cell, the messenger RNA is made in the nucleus and can sit there for several hours before it's ultimately transported into the cytoplasm. So there's the opportunity to remove the introns, which occurs in the nucleus. So prokaryotes don't have that.
Right. That's right, but the alternative argument on this is that the reason that the messenger RNA is translated immediately in the protein is a very developed end product of evolution. And not what the original prokaryotic cell looked like.
That's fine, but I'm just trying to clarify this.
No, I just want to make sure we have all of that argument.
I mean, I'm not sure I agree with that, but I wasn't talking about that.
Right. Oh, and the last thing that might need clarification is a transposon, which is a bit, which colloquially is called a jumping gene. There are actually segments of DNA that can cut themselves out of a chromosome and then reinsert in another spot. And those were initially discovered by Barbara McClintock, who I wrote about, and then were discovered independently in bacteria in the late 1960s.
That's right, although you should also emphasize for the audience that transposon is cut out of the DNA by having enzymes, by making special enzymes that cut it out and do that. Whereas when we were talking about the RNA world and introns going in and out, we're talking about self-catalyzed introns.
Right. These are introns that can actually cut themselves out without needing extra enzymes. Right? That's what made it special. Before we move on to, did you want to say something?
No, I was going to go back to the RNA world.
Please.
So the discovery of the two ribozymes by Tom Cech and Sid Altman were a huge development in the notion of the RNA world. Here was an informational molecule that could also be catalytic and cause a reaction just like a protein enzyme could. But the real, I think, fundamental thing that changed the direction of the field was the realization that ribosomal RNA, the RNA that's involved in this machine that does the translation of messenger RNA information into proteins, the RNA itself is capable to form the bond between amino acids. Before that, the thought was this complicated ribosome with 3 RNA molecules in it, lots of proteins in it, that it was very complicated and it was probably the proteins that were doing everything. Well, through a longish series of experiments, Harry Knoller showed that in fact the RNA alone is capable of catalyzing that very fundamental reaction of linking 2 amino acids together, which all life absolutely has to depend on. So here's this most fundamental process in our living world today that in fact is catalyzed by RNA itself. To me, that was the fundamental observation that said, wait a minute, this RNA world has to be real. And what has happened, I think, in the period since then is an explosion of the number of roles that RNA molecules of all sizes and shapes play in our cells today. From long RNA molecules made from what some would call junk DNA can be processed and used to control the activity of genes, small RNA molecules that are involved in splicing out introns, an incredible collection. The most intriguing to me is their involvement in a bacterial immunity system, where bacteria sense say a virus that infects the bacterium. It makes a little RNA copy of bits and pieces from the genome of that virus, stores that information away in its own bacterial genome, and next time that bacterium gets infected by a virus that has been seen before, the RNA molecule made off of the genome version that's been stored is activated, and it goes and it destroys the incoming DNA. So here's an RNA-based immunity system in most bacteria. Truly amazing.
It really
this
is.
explosion of different functions really is a real tribute, I think, to the fundamental nature of RNA. And having worked in the RNA business all along, I've always respected DNA and its storage and its permanent ability to do that, but it's pretty boring compared to RNA.
The field is getting, the RNA field is getting very exciting. When I wrote the RNA World paper, I expected people would find a series of RNA-based enzymes of all kinds. And I've been actually disappointed that so far that hasn't happened. As Ray said, the most striking RNA enzyme is the critical one that makes all proteins possible, which is, I agree, a strong reason for thinking there's some fundamental truth in the RNA World picture. I want to make another comment, though, because we talked about the introns and exons. We talked a bit, I mentioned briefly exon shuffling. That is a notion that it's related to the fact that when you look at eukaryotic genes, you find portions of the genes that prescribe specific functions to proteins, often separated out on separate exons and often reused in other genes. So that the role of the intron-exon structure, at least the eukaryotic cell, very much, there are a number of cases in which you can show, yes, it is clear that that structure has been used to combine the exons in novel forms to make new genes, which was the idea behind the role of introns as a recombination enhancer and to shuffle the exons.
I think that's true, and that's true. I mean, yeah.
It is true.
Yeah, right. Oh, there's good data.
No. I don't want the audience to be left in the impression that from our nature of our discussion, no, that doesn't happen. That does happen.
Happens,
it's a good thing. But the question of what happened in early evolution,
different,
much trickier.
It's a different question.
Related, but different. Goes back to how we made the prokaryotic genes in the first place.
Right.
And one of the great force behind the intron-exon idea is the idea you made protein genes from small pieces. And you didn't have the problem of assembling 200 amino acids into a single gene. You could find 20 amino acid pieces that might have function and then put those together later into a more complicated gene. And the argument that you never did this in the prokaryotic structure does leave you with a problem. How did I make triphosphorisomerase in that early cell?
Those of us apostates from introns early, like me, nevertheless believe what you just said, that that exon shuffling in the modern world happens a lot, and B, that genes might well have been put together from smaller pieces in the early pre-genomic stage. What we're not sure of is there's any connection between that and now, whether or not what you said.
That's right. I do think you see shadows of this in the structure of genes, but we can disagree.
Right.
Okay. I want to move on a little bit to the articulation of the RNA World theory proper. Wally, the year you published the RNA World article, that summer, at least as I read it, you got together with Jim Watson and Jeremy Knowles to plan a meeting at Cold Spring Harbor for the symposium the next year. At least that's what Jim wrote in the introduction to the volume.
Well, it's written at a time that's probably better than my memory of the theme to that.
Do you recall that meeting and what prompted it?
No.
No? Fair enough.
Name the meeting.
This is the 87 meeting?
The 1987 Cold Spring Harbor Symposium meeting on catalytic function in RNA.
Yeah.
Oh.
Huh?
Well, the symposium in 87 was on evolution, was it not? I mean, the thick one, that's where the exon theory of genes was published.
Exactly.
Yeah, okay, so
that's what I'm
right.
talking about.
That was the annual symposium.
Right.
The annual symposium.
Yes.
I certainly wasn't aware of it. I was there, and I remember writing a paper for it afterwards. I must have talked about the idea.
Well, you must have made
I always wrote papers afterwards.
You must have made an impression, because I remember I didn't go to that meeting, and I remember somebody came and told me, he said, the origin of life problem's been solved.
Is that right?
With RNA. And they were very excited about the RNA world and all this that had been discussed at that meeting. And it revolutionized if nothing else, grant funding in the Origin Library.
Very important. So, Cold Spring Harbor meetings have this kind of iconic status in the biological science communities. Do me a favor, set the stage for us all about Cold Spring Harbor, what it's like, what the symposium is, how it works.
So I was there for a long time.
Yes.
And it's an amazing place. It's on the North Shore of Long Island, and it's a sort of poor little lab set in the middle of a very wealthy neighborhood, where the wealthy neighbors invite us funny scientists over for amusement at dinner time.
This is literally Great Gatsby territory,
yes.
right?
Yes,
I mean, honestly.
It's amazing.
Yeah.
But it was really the crossroads of molecular biology during the whole growth of molecular biology. That's where everybody would come through sometime in the summer. They would run a series of courses and meetings throughout the summer. I found there, I didn't have to read the journals at all, because someone would come through and tell me whatever was happening long before it turned up in a journal.
Giving you much more time for lab work.
Real luxury.
Yeah.
And it's continued that, and it has the main feature is an annual symposium that it has early in the summer every year. It's really a command performance. If you're invited to go to that symposium, you go to that symposium, and they publish a big volume of all the things that happened at that symposium. And looking at that collection of papers is really a great history of this whole period of molecular biology from the early 60s up till now.
There's a remarkable sort of prescience to many of those meetings. They seem to know what was just on the verge of happening.
Yeah, that was one of the key things, picking the right topic at the right time.
Yeah.
That's right.
And this was one of them.
Yeah, it was a good.
Oh, well, the one actually before the splicing was announced at a symposium.
Yes.
And it was announced as a surprise.
Yep.
I was at the symposium. Yeah, those talks were a great surprise. They kept the secret. You know, they broke, and so both groups actually broke the splicing story in one evening session. I remember it at a symposium.
Now, the discovery of the splicing was a huge competition between one scientist, Rich Roberts at Cold Spring Harbor, and Phil Sharp at MIT, who had just left Cold Spring Harbor to go to MIT. So this is a real rivalry.
Yeah. So what's a Cold Spring Harbor symposium like?
What's it like?
Yeah.
Deteriorated, but
where does the real work get done? You know, what's it?
Well, it's an intimidating venue, because you know all the professionals are there. They range from people that sit in the front row and get agitated and ask questions, to Jim Watson, who would sit in the back reading a newspaper.
Loudly.
Right. And the worse the talk, the more you fold and unfold the newspaper. But I can remember Francis Crick drawing on the chalkboard is DNA makes RNA makes protein, and there's an awful lot of history in that institution.
Yes, yeah.
That's right. The messenger RNA papers were in the symposium in 1961. My first exposure there, and I suppose I didn't talk at that one. Francois Gros probably gave our paper, and Sidney Brenner gave the other paper. And Sidney is talking about the messenger RNA and saying how wonderful the idea is, and it's such a wonderful secret. And Chargaff's voice comes out from the back of the symposium. Sidney has just said it's this hermetic idea. And Chargaff shouts out, "You've forgotten that Hermes is the god of thieves." So the messenger idea was not immediately accepted by the biochemical community at that time.
This is Irwin Chargaff, a notoriously cranky and brilliant scientist at the Rockefeller. Yeah. That's a great story. So the 1987 symposium was a particularly wide-ranging meeting. There was looking through the table of contents, it's just stunning. There are a whole lot of figures who are very important people in origin of life research. There are chemists, there are biologists, molecular biologists, microbiologists, theoretical scientists. How did that all work? Watson said, again in the introduction, that he had been afraid that it would be too broad to really get discussion going. Do you remember the discussion? And was it hard to talk to? Did people form little camps, or how did it work?
I wasn't there.
You weren't there. I was certainly there. You should have. You
had a paper in it.
You should have shown me the book beforehand. I could, had I looked through that table of contents.
I have it here if you want.
I can look through it. It would probably refresh my memory. I mean, I certainly wrote a paper in it, and it was been part of all those discussions, but I can't remember it.
No, that's what the exon theory of genes was there.
That was the exon theory of genes.
And I had
the paper on
Darrell Rainey was there too, actually.
Darrell Rainey was there. Ray, you gave the paper on slipping and sliding and jumping.
My colleague did that.
Oh. Okay. So you were not at the meeting. Okay.
And one of the deceptive things about the books is that the papers are often written after the symposium.
Mm-hmm.
The most important thing is to get your picture in the front, right? Because
that shows you there.
People are there, and then they appear in the front part of the book.
That's right. The meeting is very enjoyable, but scientists rarely prepare papers ahead of time. We're different from other fields, and we talk freely, and then occasionally write it up afterwards.
Okay.
I took a bunch of those pictures, and I have some very embarrassing ones if you're interested.
I want to see those. Yeah, definitely. Here's the table of contents if you want to take a look while we chat.
Okay, you can ask him some other questions.
Blake was there too. There was quite a lot on exon intron stuff.
Yes, there was. And Ford, you had a very interesting paper about introns and hierarchy in genome evolution. And you were, as I read the paper, you were talking about evolution at different levels of the genes, the organism, the population.
Yeah, well, that's one of my hobby horses. And that's partly what selfish DNA was about too, because I mean, the theory of evolution is a very general one. It basically says if you have replicating things which have heredity so that the children resemble the parents, and they have variation between them so that some of them are different than other ones, and that variation affects how many children they go on to have, how fit they are, then you will have evolution by natural selection. So organisms are the premier example of that, things which have heritable variation in fitness, but genes do too, and that's what the selfish DNA idea is based on, and species do too in a way. Species have something like birth. They have speciation. They give rise to new species, and they also have something like death, namely they can go extinct. So that means there are some things which species have which are like traits which are selected for at the level of species. Many people think that sexual reproduction is one of those things which is selected for at the level of species, even though it's disadvantageous to the individual. It's much more economical just to give birth to children without having to have a mate. But
it just doesn't last that long.
It doesn't last. Yes, indeed, the species go extinct as a result of it, because there's not the resorting genetic information. But it would be advantageous to the individual. So that's still a hobby horse of mine, and so I thought that there is a sense in which introns speed evolution, but it isn't that organisms have introns so that they themselves will evolve. It's that species that have more introns will evolve better than species that don't have introns. So in a sense, introns are like sex. They're something which are good for the species and make it speciate more effectively or go extinct less often because it can generate innovations, but they're actually detrimental to the individual.
Now, does this go against the idea of group selection?
it is a group selectionist idea, but there are...
I always hate such ideas. I said I always hate such ideas.
I know you do.
You're just making my position clear.
But group selection, there's two kinds of group selection. There's the kind that people worry about within species, whether or not altruism is possible. That's called multi-level selection type 1. And then there's whether things like species, you know, if we believe there's a hierarchy of nature, if we believe that there are genes and then there's cells and there's organisms and there's species, different levels of the hierarchy, and each of those has something that's comparable to heredity, something that's comparable to variation, and something that's comparable to fitness, then natural selection will occur at each of those levels independently. And I believe that.
It's a very interesting idea. And you don't like it.
But so introns would be then advantageous at the higher level than the individual.
Okay.
You see, I view the introns as simply left over in the individual because they were of use in the past.
Well, both of those things can be true.
Yes.
They're not mutually.
That's right. I don't see the need for invoking group selection as opposed to just the evolutionary course that leads to the individual.
Right. You would be in the majority in that.
Okay. No, that's an accident.
George and Ray, do you want to weigh in on this question? Do you agree with this idea of selection operating on different levels?
Yeah, it sounds reasonable. Nothing. I was just thinking about, though, in the prokaryotes, we have a little RNA we're working with right now. It has a big, compared to its relatives in other organisms, it has 108 nucleotide insertion. And that insertion apparently doesn't interfere with the function, so it just gets preserved. So if you figured out a way to get rid of it, you would have discovered intron splicing, right? But the organism doesn't need to get rid of it. It's just baggage, I guess. But I mean, really,
Junk.
just kind of a thought. Yeah, junk DNA. But then junk DNA can then subsequently become useful for some reason. I mean, that's what's happened with the eukaryotic ribosomal RNAs. They're bigger, and it turns out that the extra bigger stuff is just like this prokaryotic case, an insertion. And in some cases now we discover that these insertions are actually doing something, not related, regulatory things or something.
Yeah. And what fascinates me
about
this is that, I think the popular notion is that DNA is the kind of essence of life, right? You see it in the car ads and in the perfumes and sodas and whatnot. And really, when you get into RNA, it's everywhere. It almost feels like this is really the heart of living things, huh?
Well, I think those are unfair questions. I mean, there is a set of questions which the RNA World directed at. How does life begin? How does the complicated life work?
Yeah.
What do the first cells possibly look like? How do we get there?
Yeah.
There's a sort of question in biology, how does the cell really function? How do our bodies function? Part of the answer to that is, yes, things like, RNA is a great big important part of that, as are the protein enzymes and all these things. And there are all sorts of games in cancer treatment in which people are looking at the microRNAs and saying, this little RNA, 100 bases long, elevated in a tumor, that's serious, and we should try to devise drugs that will affect that. There's another level in which our inheritance is the DNA. And that's what we pass down to our children, and that's the way our bodies work in terms of the immediate evolution.
Sure.
In fact, along those thoughts, I don't want to leave the audience with a feeling that evolution is perfect. Evolution is a process of selection, but it does not necessarily ever get to peaks of perfection.
Right.
Selection can only work in a local sense, and so you can get to local maximum. You get to regions that things work reasonably, and then you evolve something else around here, and you get somewhere else that things work reasonably. But that doesn't mean the organism is perfect in any sense.
Right.
And the organism has all sorts of imperfections, appendices, introns, all sorts of things floating around, once of use, now not of use, possibly disappearing. possibly at some moment being caught upon by evolution to do something else and appears again. The rate of mutation in our cells is determined by evolutionary processes and is in fact optimized. It's neither too high nor too low. It's a rate that seems to work for evolution, that makes evolution run at a rate that's fast enough so that the overall sweep of evolution works.
It's the Goldilocks rate.
But we're not perfect, you know, and one could imagine, and biochemists often did, you could imagine better genes, better ways of copying DNA more accurately, all that sort of thing. Turns out evolution doesn't do that.
Yeah. Maybe you could explain what would happen if evolution were too fast or too slow.
Well, we wish we knew. Sometimes too slow you can tell. I mean, the evolution is too slow, the organism cannot, the environment changes, and the organism simply cannot change fast enough.
Right. Extinction.
A species is not a unique structure. A species like ourselves, for example, contains all sorts of different types of individuals, because the genes that make up a species have a large number of variations inside that species. And so when the world changes, those individuals in a species that are well adapted to that particular future niche expand. And other parts of the species, other individual species die out, and the species, if you're looking at its overall changes in space, shape becomes taller, shorter. Global warming will affect our species. And those of us that are more heat tolerant will be in a better shape.
We need fast evolution.
And we will have reasonably fast evolution. And that sort of evolution actually is quite fast. Our species is quite varied, and we have members of the species that are tolerant to high temperatures and other members that are tolerant to low temperatures. And if the world...
We can all move to Canada.
If the road, some of us are already proceeded, you see, already established in Canada and ready for the future.
Yes, the faster the environment changes, the faster you need to evolve to keep up with it, right? Maybe we could say just a little bit about the RNA World book and how that came about. Did it grow out of the 1987 symposium?
In a sense, but it was really my colleague John Atkins and I had been involved in the RNA research for years, and the realization that ribosomal RNA was catalytic and the other the catalytic examples just made us think, wouldn't it be great to get all those two fields in one book? The early RNA evolution folks that worried about where did nucleotides come from, how could RNA replicate itself, get them in the same book with people describing all the novel new things that RNA could do, with the hope that these groups would start talking to each other more, and that by getting the folks working on the RNA World to look at the modern RNA and the intricate things it can do and the ways it can do them as a way to help guide thinking about the RNA world. We knew a lot of the folks in the current RNA world and rounded up the other folks, and there was great enthusiasm to put this book together. And then the work on RNA was evolving so rapidly that we had to put out a new edition and a new edition and a new edition. So it was great fun, actually, and I think it has had some impact on the world. But
one of the best features of the book is the appendix that shows the diagrams of all possible canonical base pairs that make 2 or more hydrogen bonds. Very, very useful.
Yeah, there are 3 or 4 appendices that are very information rich like that.
Yeah. It's one of those books if you look at the various editions, as I have done, it's clear that these were not simply just, like a lot of books, it's been a few years, the copyright's running out, let's put out a new edition. It really is a dynamic book. The volumes are different, and they really reflect changes in the field.
Yeah, I think that's what it reflects. Things were changing fast so that people couldn't just say the same old thing.
Yeah.
But I should point out that Tom Check joined John Atkins and I on the second and third and fourth edition.
Right, yeah.
The three of us.
Yeah.
Yeah. How did he get involved? I mean, I know he was involved in the, obviously in the
I'll tell you the truth.
catalysis.
Atkins and I asked him to contribute a chapter for volume 2, and he said, sure, if you'll make me an editor. But he was an obvious one to add because he had great connections in that world.
Sure, yeah.
It was a wonderful addition to the book.
One of the things I'd like to know is you managed to get both Crick and Watson, one to do a preface and the other to do, anyway, you call them different things, but you managed to get them both right forwards to the book. How did you accomplish that?
This actually may be the only time that they've published together, certainly that I know of, that they've been back to back in one volume. But they both were eager to do it. And it was so wonderful that we put those same forwards in all 4 editions of the book. But they are amusing to read. Jim talks about his early involvement in messenger RNA and ribosomes, and Francis talks in detail about the various chapters of the book and what it says about the RNA world. It's fun reading.
Yeah, it really is. Okay, in just a moment we're going to open it up to questions, but just to kind of wrap up, and we've actually touched on a couple of little bits of this, but where is the RNA world today? How has it changed the way we think about where we come from?
Well, I think it's at least provided a logical way to think about how we could have evolved this DNA world. To me, it's the most logical way to look at how it could have happened. Doesn't mean it's right. It's an idea, unproven for sure, but it's one that is approachable, and we can work at it and learn a whole lot more about the world we're in addition to maybe learning about our very early origins.
Yeah. I think that underscores the fact that science is oftentimes more about asking questions than it is about getting answers.
Absolutely.
You know?
Yeah, that's a good point.
Yeah.
Yeah.
Other thoughts?
I think, when you talked at the beginning about it being a solution to the chicken and egg problem, I mean, I think most of us do believe in the RNA world in some way. We also would agree that there are the 2 problems that George mentioned. One is that there isn't yet a self-replicating RNA, although I think most of us believe that there will be. and B, there...
You don't need it.
There is the problem of how did you get to the RNA in the first place, and was there some other polymer before that?
You guys are really good at anticipating my next question.
But mostly, I don't think people in my area care about that. That's chemistry, right? But I think it's what philosophers call a how possibly explanation. Before that, we were baffled as how it could have happened. Now, it could have happened this way, right? Whether it did happen this way, I don't think we will ever actually know. But we're no longer stymied by how could it possibly have originated. It could have possibly originated like this.
Yeah.
And I think most of us now believe that, right?
Well, yeah. But the question of the RNA replicase, the alternative is a protein replicase. Right. And we actually have proteins that can replicate RNA. They're called polymerases, right? And so the fundamental question is, if you have a ribosome early on that's making peptides, how complex does that peptide have to be to become a polymerase? And so while we're searching for the RNA replicase, maybe we should be searching for the peptide replicase. Now the problem is, experimentally, it's much, much more difficult to develop, create random peptides and assay them and stuff. But
that's why you're going to do it.
No, I'm not going to do it.
It could be done. We have the technology to create random peptides.
Another thing which hasn't been mentioned, which it probably should be, is the work of Mike Yaris. And he has actually, under prebiotic conditions, created very, very small RNAs of 4 or 5 residues, where one can catalyze the attachment of an amino acid to another. These very, very small things. And I think he's even showed that you can get a few of these together and you can even make a peptide bond. So I don't know that your original RNA doesn't necessarily have to be 200 residues. You get interesting things happening with much smaller RNAs.
One interesting aspect of this is that we're starting to see where research on the origin of life 4 billion years ago starts to intersect with the very cutting edge field of synthetic biology, trying to create life in the laboratory. And the information that's being gleaned out of this research is feeding into some very high-tech, biotechnology.
Mm-hmm.
Yeah? Well, thank you. I think with that, we should probably open it up to questions. What we're going to do is we have 2 microphones here and here, and you can just line up in front of the microphone. If you have a question, feel free, stand up, and please just come up and line up, and we'll just take you as you come.
Yes, my name is Ronald Wilson.
Yes, thank you. If I may, as Ron did, please do identify yourself when you come up. Thank you.
Yes, my name is Ronald Wilson. I work for the federal government. I do some research on origins of life, and you all did touch upon synthetic creation of life in the laboratory. And I was wondering, based upon what you all know of evolution, natural selection, and also RNA and DNA, do you think it will ever be possible or feasible in the future, whether it's near term or long term, to actually create life in the laboratory?
That was a nice segue.
Yes.
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 Yes. Jack Shostak is going to do it soon, right?
Well, he's well
You think,
along.
Jack?
It means
Yes, I think so. I mean, there's a great,
Listen,
again, in some ways, it's a question that depends on our definition of what is life.
exactly.
To the extent that
Okay.
I'd be delighted to see a RNA molecule that can copy itself and continually copying and mutate to better things, to do that process better and better, I would consider that an interesting first step on an evolution of life. To actually get that RNA molecule into a cell and self-sustaining, that's still harder. Yes. We don't even have that very first one yet, and we don't know where the precursors to that really come from. On the other hand, I'm quite confident that we will eventually get there, that we will get a model organism that can evolve, replicate itself, evolve to better things. To actually create an organism that looks like a bacteria or looks like the original cell is going to be quite difficult.
I can't resist.
Again, that turns on,
There's an alternative.
the speed of evolution.
You said laboratory, and I'm not sure what your definition of laboratory is, but there is a whole other possibility of artificial life, and it's with computers.
Yeah.
Right? I mean, there's a lot of
that's a different
exciting,
form
the
of life.
possibility of a self-aware computer of some sort is not, it's being thought about and talked about. And so that's another life, right? This artificial life or something. It's just not in the laboratory.
How would you know it's self-aware?
Well, that is a major issue in the whole discussion, actually, is what is self-awareness?
You ask.
Yeah.
And if it answers,
Yeah.
How do I know I'm self-aware?
how do you know I'm self-aware? That's
But that's really being very actively discussed and taken seriously.
Yes. Okay. Yes. Hi.
Hi, Tom Barrett, and I hope this question's not unfortunate, but from the perspective of these early RNA replication environments, we know that pH and solute concentration is extremely important. Do you envision some kind of enveloping situation occurring around the nucleic acid, or this just being a free environment, like perhaps a pool of water? Certainly not the ocean, I wouldn't imagine, but what are your thoughts on the actual physiological conditions surrounding the RNA early in replication?
Well, it's actually a very difficult question. I mean, there's a sort of simple answer in which I think in the 86 paper I imagined little puddles of water and doing that. And Dan Jay then got inspired to say, well, maybe the first membranes will be positively charged polymers that you can then use to wrap nucleic acids. And we even did a little experiment to show, yes, it makes a little sense. I do fundamentally agree that as to have something that we're going to call any sort of proto-organism, it's going to have a boundary, some way of separating and permitting the improvements that develop in the replication to pass to daughter organisms. You're going to have some sort of structure. But I think the ideas about the structure are at least as primitive as the ideas about how to actually make the self-replicating RNA molecule.
Harold Morowitz over at George Mason would say you have to have a membrane.
Well, that's part of what Jack Shostak is doing now, is trying to get things that replicate within a membrane structure. So that's why I'm putting my money on him.
Okay.
I would too.
Yeah? Okay. Yes, hi.
Hi, my name's Mary Fraker, and when I first started listening to this, I was wondering, well, what on earth good is DNA? I mean, RNA is doing everything. And is it because, and so why would it still be around if it wasn't doing anything? But is it because it's so capable of it's like a library, the genomes could get longer and more complicated and we could all be here talking because the DNA was able to store that much information? And then my other part of the question is, were any of you in the RNA Tie Club?
I'm afraid the answer is we weren't in the RNA Tie Club. We're all a little later than that group.
Yeah.
Your question is absolutely right. I think our conception of the role of DNA is completely that of a much larger information store, a much more accurate one.
Yep.
We know of a large, DNA today uses a large number of error-correcting devices to preserve the information. And where we have RNA genomes functioning in viruses today, they are not as accurately copied. They have about a 1 part in 1,000 error rate, whereas the error rate in DNA is down around 1 part in 10 to the 6th, 1 part in 10 to the 7th. And the specific, there are lots of specific reasons for that and detailed models for how that actually happens.
But Wally, RNA could have evolved, might have evolved.
It might have evolved to do that. We do not.
Those same error correction mechanisms.
But it is intrinsically less stable.
Right.
Chemically.
So it is
intrinsically. We don't know whether you don't know what's cause and what's effect. In the sense, you don't know whether. Yes, you could have evolved it to be more to have the error correction mechanisms, and you could have evolved a pH balance in the cell keeps the RNA stable.
Yeah. But certainly that one hydroxyl group difference makes a very big difference in the chemical activity of the molecule.
Yes.
It's a great question to be asking in the Library of Congress, though. I think this is.
Yes, DNA is a fantastic library.
Yeah.
And
its very inertness is part of the value, right?
There's now an effort to look at storing book information, a whole book in DNA sequences.
Yeah.
You synthesize DNA in any sequence you want. You can do that now. And you can encode all the information digitally that you're interested in doing into this 4-letter code and store it away someplace where it's stable and lasts a long time. And then when you want it, you just read it out and you get the book back. So this is
if you put enough copies down. I mean, we have managed to read DNA from 20 million years.
Yes.
Occasionally, at least 1 million years, at least longer than books have lasted so far.
Longer than papyrus.
Like cave painting.
Well, cave painting is 30,000 years, but I was thinking of, we have read the Neanderthal sequence, which is probably 100,000 to several 100,000 years, and we have occasionally, I think we covered plant genes, if I remember correctly, and I think that's about 20 million.
Yeah.
Although, as I said, lots of copies, because lots of damage gets done over that time.
You need redundancy.
Yeah.
Okay, yes, hi.
Hi, Aparna Kishore. Dr. Gilbert mentioned something that I was curious about, which is the role of viruses in the interplay between the evolution of an RNA genome or the conversion from an RNA world to a DNA world. I was wondering what your thoughts on this might be.
I was talking about introns. I'm not sure I talked about viruses. Trying to think what.
You didn't. She wants you to.
Oh, what? You want me? That's what
Good for you.
I'm not sure I can say anything useful. In some ways, on this scale that we're talking, the viruses, I'm happy to talk about bacteria as highly evolved, streamlined organisms. The viruses are even better evolved, streamlined organisms. The one place in which they play a role in our genome is actually the thing that you were mentioning. The RNA viruses provide sources of DNA that then, copied back into DNA, provide sources of DNA that then are in multiple copies in our genomes. But that's mostly a later phenomenon, I would think, in evolution. So I don't have anything to really say about early evolution.
But doesn't this also bear on lateral gene transfer?
Well, people always ask about viruses and the origin of life and the tree of life, and it's really not clear what you can say about that, because it seems, I would guess, that most modern viruses are relatively modern evolutionary inventions. But that doesn't mean that there couldn't have been virus-like things in the past, and almost certainly, in an RNA world, and in fact in vitro evolution shows this, if you ever did get a self-replicating, an RNA that could replicate itself and other RNAs, then other RNAs whose only function was to be replicated by this replicase, but which could not themselves replicate, would evolve, right? I mean, the parasites.
Yep.
And that sort of thing happens all the time, and those would be analogous to viruses. So presumably the RNA world had things whose only goal in life was to have copies made of themselves by the replicase that was doing the real work of keeping life going, and they would have been viruses, the equivalent thereof.
Yeah, in fact, let me make a comment that may not be immediately obvious to everybody. When we think of replicating RNA, we're thinking of molecules that might be a few hundred bases long that are sufficiently complicated to copy themselves. Then if we want to make more complicated RNAs, we might think of getting to molecules that may be 1,000 bases long. And when I talked about RNA introns moving pieces of RNA around, I'm talking about sort of conjectures that you have molecules maybe 1,000 bases long, and you've got 20 base long pieces separated by 100 base long introns, and you're assembling those 20 base long pieces into interesting molecules that are a few hundred bases long. That's one of the reasons for that sort of size range turns out to be this error rate in the RNA copying. If I get to a molecule 1,000 bases long, I can't actually copy it exactly. It's going to always have an error in it. That means that the organisms that we're talking about, when we begin to talk about the first organisms that are inside a membrane, have a number of different 1,000 base long RNAs in it. They've got 10 or 12, maybe 50 different pieces doing different things, being copied, becoming enzymes, doing all sorts of things. Lots and lots of little pieces that all have to be held together, and if too many of them get lost, the organism doesn't work. And when we change the DNA, we begin now to talk of structures that are 50,000, 100,000, million bases long.
Right.
It's wildly different structures. And we see this in the viruses. We have RNA viruses that have 10 or 12 genomes in it. We have DNA viruses that have much larger genetic structures. We see it all around us today. And it influences the biology of the viruses in all sorts of ways, in how recombination works and all this sort of thing. So.
Okay.
The next question?
Yes.
Yes, my name's Timothy Thomas. You presented what to me was a new idea, the idea that introns might be a kind of an analog to sexual reproduction in the sense that it accelerates variation across the generations. So I'm wondering about the role of RNA and DNA in sexual reproduction, and what you think about the fact that eukaryotes contain both intron scrambling and DNA and sexual reproduction.
That's a question for you, Wally.
Okay.
Yes.
Generally, the time scales are different. The time scale of sexual reproduction is the immediate mixture and separation of the genes from the parents into the offspring in the next generation. And one of the arguments for why sex works so well is that the new combination of genes prevents parasites from learning how to get to the parent and get to all the offspring rapidly, because we're constantly changing the pattern of genes in the offspring. The recombination also takes place during sexual reproduction, and it also produces novel gene combinations that are in the offspring that are not in the parents. Not that many novel combinations. 50. What in our case? 46, I guess. 46 novel.
Close enough.
Close enough. 46
At our age,
novel combinations.
doesn't matter.
Plus the number of mutations. Whatever the right number is in humans, 200 per generation or something of that. I have to think about it to actually remember the number. But the recombination process inside a gene that makes really novel combinations occurs in a given gene in a much longer period of time.
Yeah, timescale way different.
So.
Hi, I'm David Grinspoon, Planetary Science Institute. It's been a really fascinating discussion. And to bring it more explicitly back to astrobiology for a moment, in 1977, we didn't know if there were any other habitable planets. Now we know that there are perhaps 40 billion in our galaxy alone. So imagining that some fraction of those might have carbon-based biospheres, I'm curious if you guys picture an RNA world as a sort of inevitable phase that those biospheres are likely to have gone through, or most of them, or some of them, or is it more a fluke, potentially, of terrestrial evolution?
Universal. You know, if you want, I mean, in fact, all of us, I think, in this discussion of the origin of life, I think we all believe that if we can, in fact, duplicate it in the laboratory, and the RNA world is one model of how you might duplicate it. We don't know it's the right one, but it's a model. It is a model which, when we think about it, we would think it gets used for an origin of life everywhere. And I agree with you. We think there are very large numbers of probably Earth-like, probably carbon-based, you know, roughly the same chemistry, water and all that. Very many examples. I don't know, 40 billion is a perfectly good number. And one would think that the underlying chemistry and physics is the same, and therefore an RNA world, which is a way of getting a life code, is a way of proceeding. Whether that leads to the same genetic code is an interesting question, and there's all sorts of arguments about what is the real structure of the genetic code. Is it an accident, or does it have a deeper structure that's related to the properties of the amino acids as they finally come out? We certainly, I think, all believe it's the same RNA molecules. It would be the same amino acids. Whether we think it would all be left-handed amino acids is a different argument. You know, we might easily think, oh, that's an accident, and everything would be different. And I suppose my own prejudice is it's probably an accident. I don't know of a physical reason why the sugars that we use and the amino acids we use have a specific handedness. Although, as I was mentioning earlier, one of the mysteries in physics is that there's matter and not antimatter, and which we don't know a reason for. So there could be a deeper reason in the amino acids that we do not know, but is related to the weak energy and things like that. I mean, every now and then somebody tries to conjecture that the asymmetry and the weak interactions is the reason that the amino acids are left-handed. Yeah, one doesn't know if that's true or not.
Yeah. That's why what we say, or we should say, is that we're trying to understand the origin of life as we know it, as the life here. Now, it's your job as the planetary guy to go out and find us another life system. So if you can find us a second independently evolved life system, then we can compare them and say, well, see, lots of things are being shared by the independent origin or not. Right, we're gonna just need more money, right?
Yeah. That's right. And unfortunately, if you find the same one, then we get in this panspermia argument. Yeah. We still don't know the answer.
But it might be risky to look just for RNA. There are folks that think there's something else that came before RNA that evolved into our RNA-like world that we imagine.
Yeah. Well, David McKay thought he had found life in the meteorites from Mars, right? And one of the main criticisms of his fossil organisms were so small they couldn't hold numerous ribosomes that you would need, okay? But it might be, maybe they're living systems where they don't use ribosomes to make proteins. Maybe they do things very differently. And of course,
maybe it's just the RNA world molecule, the RNA world organisms. They
maybe.
Right.
Can they further?
Right. But so if there's so much prejudice based on what we know about life here as to what we're going to find somewhere else, but we really need that second system. So even if you go to Mars and you find life, the hope would be that it's independent origin as opposed to just lateral transfer between the planets.
And it's one of the reasons that in looking for life on Mars, we shouldn't look for ribosomes that are like ours.
Yeah, exactly.
There are some efforts beginning to look for RNA world organisms that might still be extant on Earth, and looking at small water pockets in deep mines, and extracting RNA from that droplet of water, and looking first for ribosomal RNA. If you find ribosomal RNA, it's not interesting because we're looking for the really early one. But any RNA that you can sequence and where you don't find ribosomal RNA is going to be very interesting. It's just being looked at some low level. But it's cheaper than going to Mars.
Is it clear to everyone why, if you find ribosomal RNA, it's not interesting? Do you want to go over that just briefly?
Well, if you're...
It might be interesting.
Go ahead.
It might be interesting.
Sure.
But if it's not using ribosomal RNA, it's definitely interesting. Okay.
Because presumably it would be before
It'd be different.
the
Right. Be
ribosome
different.
had evolved.
Be
right?
different in some way.
Okay.
Yeah.
Thank you.
Thanks.
But then you'd have to show that it's not a virus, too,
Right.
I guess.
Yep.
Okay. Thanks.
The sequence would tell you a lot.
All right. This will be the last question, and we'll wrap up.
Okay, thank you. So my name is Daniel Ben-Alevi. You just commented on something that was missing to me. Maybe you can talk a bit about the connection between the RNA world and the genetic code. So after the genetic code was discovered, there were some papers that they discussed the fact that similar amino acids are encoded by similar codons. And they say, they summarize that if it would be more, might be adaptive, that if you have a mutation, then a similar amino acid will be translated and there will be no damage. But assuming there was an RNA protein world, the sequence of the RNA should not just encode for something hereditary, but should also encode for a function. So certain nucleotides might have maybe related to certain functions. And maybe you can elaborate. So maybe it can help to find today evidence for the RNA world, because one example I can find is that sugars are always activated by uracil, and the translation, the energy comes from GTP, and for other, so you have examples in the cell that specific nucleotides are involved in specific processes or in specific compartments. So, yeah.
Wally.
One's thinking. I think one's thinking about how one gets from an RNA to world in which, let's say, we have cells with RNA and RNA enzymes and RNA being duplicated, and we begin to make polypeptides. That requires a process in which we gradually build up some way of recognizing the amino acids, some way of stringing them together, and some way of dictating that order. The final result of that is a genetic code, a messenger, and a translation molecule that recognizes the amino acid today with an enzyme, in some previous instance without an enzyme, and ties it to a message. It's not clear at all how that process occurs. There's no way of thinking that the enzyme, that's the RNA enzyme that's being duplicated by the protein enzyme, are related. They presumably arise separately, and one replaces the other.
Yes.
the conjecture about the code, one of the conjectures is what, as you mentioned, that there is some structure in the, there's several possible conjectures. One type of conjecture is there is something about the codon or anticodon that's related to the amino acid. And so as you begin to develop triplet codons, or triplet anticodons, they pull out the appropriate amino acid. Another argument is, no, that's not the way it happened. You develop a codon, but you use the mutation structure to tie together related amino acids. And you, as I mentioned, there are arguments about the code. There's a paper called The Code is 1 in a Million, as I remember. It's a very amusing paper, and it tries to go in detail in trying to argue that the structure of the code has certain features that make it look as though it's not a random connection between amino acids and codons, but there's all sorts of interesting structure that's related to things like ability to withstand mutations, things like that. But as the comment I made, you know, the code can't be perfect in the sense that you have to have mutations. Things have to change. You know, there's all sorts of error-correcting devices in the amino acid today, in the amino acid recognition. There's error-correcting devices in the enzymes that look at the amino acids. You know, today's structure is a very complicated one that's full of detail. It goes beyond just the raw biochemical. You know, messenger makes protein, and there are all sorts of features that are involved in how do we actually make proteins accurately enough to work. And some of that's even tied to the issue that proteins can't be too big, because the error rate in the protein finally begins to overcome the function of the protein. If you're better off making proteins in moderately small chunks and putting them together to assemble large structures.
And the several hypotheses are not mutually exclusive either, so...
No. None of this is known well enough. But for fun, look up some of these papers on the structure of the code. There have been a number of people trying to conjecture things about that.
Yeah.
It's a cottage industry of some...
Yeah, it's amusing industry. But then the papers are fun.
And the concept of the RNA world, the addition of the code is probably relatively recent, even though it's obviously extremely ancient. But your 50s particle, the ribosome has a 50s particle and a 30s particle. The decoding occurs in the 30s particle. You can completely eliminate the 30s particle, and the 50s can still catalyze peptide synthesis. Okay, and so the whole catalytic part of the ribosome doesn't need the messenger RNA, doesn't need the code. So, so you can make polypeptides. They obviously would be random, and that's another matter. But all things considered, looking at the evolution of the ribosome, the coding part is, in the grand scheme of things, a relatively recent phenomenon.
Good point.
Well, it's all, I mean, the great danger in all of this is we look on these in the wrong time frame. We're used to thinking of enzymes as working quickly, and you know, and the cells as well designed as they are today. And when we're going back in this image of early evolution, one's trying to really think of something in which the organisms slowly grow, very slowly, but fast enough for the RNA not to decay. I mean, there's all sorts of problems that hidden away in this. Last question.
I've been told we can have an encore question.
All right. So last question, considering we're in DC. Have any of you ever had discussions with policymakers or government officials about the origins of life, and especially with those policymakers who may have different views on this topic? And if not, why?
Good question.
It might be useless.
Have you?
It's a deeper problem there.
Well, my postdoc sitting over here, Madan, just was here for such discussions, and he just came to this meeting for fun, but he was actually here for such discussions with what? Some congressman?
Yeah, I just speak to some of the congressmen representing Texas. They are not at their best, but I'm not sure they understand.
Well, thank you for trying.
Thank you.
I appreciate it.
This has been a presentation of the Library of Congress. Visit us at loc.gov.